In this video, I will be taking you through the principles of targeted therapy. In previous videos, we talked about and introduced you to the medical oncologist and how they play a role both in the localized and locally advanced setting and in the metastatic setting in administering systemic therapies, principally chemotherapy. We went through their background and that they are often the central coordinator of care in the setting of cancer, and that they administer and are experts in treating with systemic therapies that are usually administered IV or given orally. We talked about in the previous video about chemotherapy, and in this video, we'll be focusing on targeted therapies. To do so, though, we really need to go back and visit the how and why we get cancer, where we discussed the central dogma of biology, and that this entails all cells having one copy of DNA that is identical in each cell, and that this DNA, through transcription and then translation, expresses itself in proteins that are the functional component of the cell. These proteins are each determined by the specific sequence of bases in the DNA and then the RNA, and that the genes can be turned on through transcription and then translated into protein. We call this expression of a gene. So remember, we have many, many genes in every cell, but certain cells will only turn on or express some of those genes. So stomach cells will express stomach genes and their proteins. And then we talked about how cancer has abnormalities in the DNA, either the sequence or the structure, as I'll show you, that then leads to abnormal expression of oncogenes and underexpression of tumor suppressor genes. We also talked about how cells normally divide and are told when to, and are often quiescent and not dividing. In cancer, because of these genetic changes, the cells are always being told to grow and divide. With this central dogma of biology, it's the protein, again, that is the functional component of these DNA changes. And the protein structure is a sequence of amino acids, the building blocks that, again, are determined by the DNA code. In triplet DNA bases will then determine what amino acid and sequence. This specific sequence of amino acids, because they all have a slightly different three-dimensional structure, will then dictate how it folds on itself. And these folds are important because they form crevices and other structures that then lend to their actual function. We also showed how different genes can encode different proteins that are, in this example, shown by blue, red, and green, 3 different gene products that then can bind to each other through these crevices and three-dimensional structures to create a megaprotein or quaternary protein structure that then has specific functions. Again, when the DNA changes, amino acids change, the folding may change, and the function may change. And when those changes are in an oncogene or an oncoprotein, usually that lends it to become overactive in its function, and tumor suppressor genes often lose their function through these changes. The consequence ultimately, again, is that these cells are abnormally dividing and growing. We leverage that knowledge by talking about chemotherapy, which targets that actual downstream effect of dividing and growing. And we reviewed that in the previous video of all the different kinds of chemotherapies that can be administered that all essentially will get into and inhibit DNA synthesis and then cell division, which is required by a cancer cell. We noted, though, that of course normal cells need to divide when appropriate, and so therefore that's why chemotherapies can have side effects, because we are inhibiting and having effect on our normal cells. Just to note again, we have a number of recognizable chemotherapy drugs and their combination regimens that we went through. So now, though, what about if we learn about in an individual patient's cancer which oncogenes are overactive and their proteins, and can we design therapies to those specific proteins that are overactive? As a review, again, there are 4 main types of genetic alterations that we need to be aware of in order to understand how targeted therapies work. And a brief review here, remember we have 23 chromosomes, 2 copies of each one from each parent, and that each of these chromosomes has a bunch of genes on them. and there'll be 2 copies of every gene, one on each chromosome. And so in the normal setting, this as a normal chromosome, if this is our gene of interest, different genetic alterations can occur that then are oncogenic. The first main one that we talked about is copy number alterations, where you can have gene amplification. So instead of 1 copy on every chromosome, you can have many. I've seen 200 in some cancer patients' tumors. And you can also have deletion of large components or chunks of the chromosome, which would then abolish that 1 copy of the gene. You also have a very common scenario where you can have a single base change, single base pair change that then leads to a different codon, that leads to a different amino acid, that leads to different function, which can be either oncogenic, like overactive of an oncogene, or can be loss of a tumor suppressor gene's function. And then we can have small insertions or deletions of DNA that then negatively affect the protein. Usually this leads to loss of tumor suppressor function. So in this case, we have additional DNA that's inserted that then leads to what's called a frame shift mutation that then alters all the downstream amino acids and changes the protein function, usually loss of function, or you can have deletion. And then finally, a little bit less common, but occurs occasionally in gastroesophageal cancer, where you have a fusion and the chromosomes fuse inappropriately and create new oncogenic proteins that are abnormally signaling where they would not be if, in the absence of that rearrangement. So with these 4 genetic alterations, this leads to the oncogenic problem. And as these genetic events accumulate in a given cell through various means, remember we talked about how different things can change the DNA sequence, eventually the cancer cells will become immortal and be able to continue growing without dying, and as a consequence of each of these genetic problems, which are often several in a given cancer cell. We talked about the heterogeneity of gastroesophageal so far anatomically, looking at the esophagus, squamous cell versus the esophagus adenocarcinoma and G-junction versus the stomach. And we looked also under the microscope that there are a number of different histologies that a pathologist can report that they're seeing, like intestinal type, diffuse type, mixed type, etc. But when we do molecular profiling, which entails looking at the DNA sequence and the structure and the expression of different proteins, we see immense heterogeneity from one patient's cancer to the next. That said, we can cluster them to be within specific categories, and that's shown here. And so the dark red is talking about esophageal squamous cell cancers, which are really a different cancer. I've mentioned several times through the videos that their genetic profiles are often different, their molecular profiles are often different. The adenocarcinomas are encompassed in these 4 different main subgroups. And these 4 main subgroups, we've talked about some of them, MSI-H or deficient mismatch repair, which occurs mostly in the stomach, and genomically stable subgroup, which entails a lot of the diffuse type histology, signet ring. We talked about CDH1 mutation. We're going to talk about Claudin 18.2, and in the rare scenario where there can be a fusion of that gene. And then Ebstein Bar Virus is another less common association with Ebstein virus infection. And then finally, the most common, as you can see in purple, is the chromosomal instable. And so although we recognize that there's a lot of heterogeneity in gastroesophageal adenocarcinoma, you can see that a large majority are this chromosomal unstable. And the majority of the gastroesophageal junction, the esophageal and G-junction and cardiac cancers are chromosomal unstable. In this figure, what you see here is a number of common oncogenes and 1 tumor suppressor, P10. Across the 4 main classes, also known as the TCGA, the Cancer Genome Atlas classes. And imagine each of these columns, different patients' tumors, and they're clustered by their similarity in molecular profile, their genetic and protein expression profiles. And what I want to point out is that you can have amplifications of genes, mutations of point mutations of genes, or deletions of genes in blue. And usually deletions will be in tumor suppressors. So that's why the blue is down in PTEN and not so much in the oncogenes. And you can see where amplifications occur is that they really are a hallmark of chromosomal instability. Chromosomal instability meaning they get structural changes in the DNA and you get extra copies of genes, oncogenes specifically. And this is pertinent again to our disease type, which is predominantly chromosomal unstable in a large proportion of the tumors. HER2 gene amplification, which is one of the first targeted therapies approved across cancers and the first one approved in gastroesophageal cancer, makes up about 10 to 15% of all gastroesophageal cancers, and you can see they really cluster in chromosomal instability. There are a number of other genes that are amplified: EGFR, FGFR2, MET, that we're going to go through here in a moment, because they are also pertinent in this disease, and together they make up almost 30 to 40% of the disease with just those four gene amplifications. So what are those genes? Those genes are called kinases, and we have about 500 or so kinases in humans. And what is a kinase? A kinase is a protein that's encoded by a gene, but it's a special protein called an enzyme. And an enzyme, in this case, is one that's catalyzing the transfer of a phosphate group from ATP to a specified molecule. So it is basically catalyzing a chemical reaction. And this map here is showing a whole bunch of kinases that are grouped into different subgroups by their acronyms. So what we are most interested, because each of the ones I've already mentioned are tyrosine kinases. Tyrosine kinases are often abnormal in cancer. And to blow those up, we can see that all of the ones that I just mentioned are now highlighted: HER2, EGFR, which is also known as HER1, HER3, we're going to talk about in a moment, the VEGF receptors, vascular endothelial growth factor receptor 1 and 2, FGFR2, fibroblast growth factor receptor, and the MET and RON tyrosine kinases. So each of these are going to be important as we'll go through the remainder of the lecture and learning about targeted therapies. This branching is really indicating how similar each of these different kinases are to each other. And so they all have similarity to each other, and that's why they're grouped as tyrosine kinase as opposed to a different group like, say, tyrosine kinase-like kinases. But these ones are all similar to each other, and that also will become relevant when we're talking about targeted therapies and how specific they are to the intended target. So each of these circled tyrosine kinases that I mentioned on the previous slide are known as receptor tyrosine kinases. They are kinases that span across the membrane of the cell. So there's parts of the protein that are outside the cell and parts of the protein that are inside the cell, and some that are inside the membrane. And the way that each of these receptor tyrosine kinases work is that in the inactive state, they are not active. They're not transducing any signal. But each of them has specific ligands that bind to the outside of their component and the extracellular component and bring these two receptors in vicinity of each other and activate the intracellular tyrosine kinase domain, the active component of the enzyme. And that activation then leads to a signaling cascade. So the kinase domain is on the inside of the cell and as part of the protein. And when they're activated, it leads to a signaling cascade where it activates other proteins that then activate other proteins, etc. And there's a downstream signaling cascade. In this case, with receptor tyrosine kinases, each of these usually are telling cells to grow, proliferate, divide, and to survive. So we'll use an example of one of those receptor tyrosine kinases, MET, to really get an understanding of this. And remember, MET is an example of an oncogene that in the normal setting, in a normal cell, is being turned on and off appropriately when the cell needs to grow, when it needs to divide, if there needs to be migration of cells. So in cell development, in embryos, for example, MET is highly upregulated. There are also tumor suppressor genes in our body that are keeping each of these oncogenes in check. They're telling the cell not to grow, et cetera. And so there's a balance of these genes being on and these genes being off, and an interplay between them when the cell needs to be doing each function. So when we look at a cell and its nucleus, in the example of MET, which is a protein, a receptor tyrosine kinase that spans through the cell wall, when the ligand, in this case called HGF, binds to MET, it brings the two receptors in close vicinity, and it causes receptor homodimerization because the receptors are the same. So it's homodimerization. And in the inactive setting, there's no ligand and the receptors are not in vicinity, so there's no signal transduction. However, when we do get the ligands, they come together, there's a conformational change of the protein that then leads to a signaling cascade. We should also note that not on this figure is not highlighted is there are tumor suppressors that are present that are inhibiting each of these proteins along the way. And again, this is all kept in a delicate balance. So all of these oncogenic proteins are able to transduce a signal into the nucleus when appropriate and will lead to proliferation, other features like mobility and invasion unique to MET, and survival of the cells. On the other hand, if the balance is tipped in favor of the function of tumor suppressors, this will all be turned off. There's no ligand around, and the cell will not be doing these functions. In the setting of cancer, when you have activation inappropriately through those genetic changes of the DNA of any of these oncogenes, including MET, then you will have a balance tipped in favor of each of these functions because the cells are continually signaling through these pathways. As I mentioned, amplification of genes is a very common scenario and particular to chromosomal unstable subset of gastroesophageal adenocarcinoma. So what happens in that setting is that we have way too many copies of the gene of interest, in this case MET, which leads to way too much protein transcribed and translated, and then we have more receptors at the membrane than normal. And this occurs in about 5% of gastroesophageal cancer. Now, if you were to sequence the gene itself, it would be a normal sequence of the DNA code. There would not be any mutations, but there'd just be too many copies of the gene. That's what gene amplification is. And so when you have all of this extra protein at the receptor, these receptors start to bind to each other, even in the absence of having ligand around, because there's just so much of it. And that leads to a constitutive overactivation of this signaling pathway. Ultimately, this leads to cell division, growth, proliferation. And again, this is why we used chemotherapy downstream, was because it didn't matter what the genetic event upstream was. Ultimately, the consequence was that the cells being told to grow, the definition of cancer, and divide, and chemotherapy would come in and inhibit at this step. Now, this signaling pathway, as you see here with amplification of MET, is not unique to MET. You could literally exchange the name to HER2, and HER2 works the same way. This is a normal stomach cell with a normal amount of HER2 at the receptor, but in the setting of amplification of HER2, where you have too many copies of the gene, you get too much protein, you get too much activation of HER2, and often these receptors at the membrane, like MET and HER2 and others, use the same downstream signaling molecules, RAF, RAS, MEK, ERK, etc. The same downstream molecules then transduce that signal. Again, there are a number of receptor tyrosine kinases that can be amplified inappropriately in gastroesophageal cancer, and you see their incidences here, HER2 the most common, but there are others as well. MET, we've talked about, EGFR and FGFR2. There are abnormalities that can occur, as I mentioned, in any of these proteins that are listed here. RAS is a very common gene and protein product that's abnormal across cancers, including this type of cancer, which it can be mutated and overactive, and or can be gene amplified, which is actually more common in gastroesophageal cancer. So again, doesn't matter which of these is abnormal, and often more than one of these can be abnormal in a given patient's cancer, but the ultimate consequence is that you get overactivation and proliferation of the cancer cell. So again, that's why chemotherapy was used before a lot of this was understood, because we knew that these cells were dividing and that chemotherapy can come in and interject at that step. But can we leverage the understanding of any of these oncogenes and their proteins that are overactive, and can we design therapies to inhibit them at that step? So that is the difference between chemotherapy, which was working to inhibit growth and cell division, and targeted therapies where we could identify in a given patient's tumor in a personalized manner what the genetic events are, and then assign a therapy that is directly trying to inhibit that particular gene, something that's referred to as oncogene addiction. So in a HER2 amplified cell, that cell is addicted to the HER2 signal. So if we can knock out that signal at the level of HER2, can we have more specific inhibition and therefore also have fewer side effects compared to, say, chemotherapy? This is not always true, and as I'll show you in a moment, but that was the concept, was let's make this more specific to a given patient's tumor in a targeted manner. So this slide is an important one because it now introduces to the main types of targeted therapies. And the first thing to understand is that there are really two main classes: antibodies and small molecules. Antibodies are normally part of our immune system. They're made by specific white blood cells called B cells or plasma cells. And antibodies are proteins that are encoded by a gene and expressed. And there are parts of the antibody that are constant and the same in every antibody in a given person. And then there are parts that are variable, that are different in every antibody. And the way these antibodies work is that, like a lock and key, a specific antibody will bind to a specific part of a molecule, whether it be a protein or other. And they recognize one specific epitope times two, because there's two parts of every antibody. These drugs are usually administered IV, and that we can make these antibodies to mimic what our own immune system is making, but make a whole bunch of them specific to a given target. So think of antibodies as the lock and key, where we have millions of locks and millions of keys, different sites that they can bind to, called epitopes, throughout our body. So these antibodies can bind to and inhibit infectious agents like bacteria and viruses and neutralize them. They can bind to cancer cells, specific parts of a cancer cell or protein, and inhibit them. And that's the idea of what we're doing here. And a classic example is an antibody, trastuzumab, to HER2. Antibodies that are manufactured and treated as drugs end in AB, antibody, monoclonal antibody. Monoclonal means it's specific for one site. We can also engineer these conventional antibodies to be a little bit more interesting, in that they can recognize two different epitopes. So you can change it so that one part of the antibody inhibits and binds to a certain epitope, and the other one binds something else. And we'll give an example of that later. We can add chemo drugs to the back end of the antibody, and this would be called an antibody drug conjugate, where the drug is attached to the antibody, and it can act like a smart bomb because it will bind to whatever it's designed to bind to and deliver the chemo directly to the cells that have that, potentially sparing cells that don't have that protein from this side effect of this chemotherapy. That's the concept. And an example of that is trastuzumab-deruxtecan. In contrast to antibodies, small molecules are chemicals. They're chemicals that can be made. And an example of a class of these inhibitors are called tyrosine kinase inhibitors that we're going to talk about. We mentioned what a tyrosine kinase is. And we try to make them specific for one target, and some can achieve this, while others are a little bit less specific, and we'll get into the reasons why that is in a moment. An example, one of many, is afatinib. Any drug that ends in IB is a small molecule inhibitor, and this drug is an inhibitor of both HER2 and EGFR. Again, these are chemicals that are it's synthesized and designed by biochemists to inhibit specific molecules in the tyrosine kinase domain inside the cell, as I'll show you. So that another major difference between antibodies and small molecules is their size. Antibodies are massive in comparison to the size of these small molecules. And the relevance of this is that these antibodies can't get inside cells, and they therefore act on the outside of the cells. Proteins that are external have an external component outside the extracellular domain. And I showed you how receptor tyrosine kinases stick out outside of the cell, and that's where these antibodies have proven most useful. In contrast, these small molecules can get into the cell and work on the inside components of the cell. And I'll show you that in a moment as well. So an example then of a pathway that's commonly abnormal in gastroesophageal cancer is the HER2 pathway. And again, HER2 gene amplification leads to a lot of HER2 protein at the receptor level at the on the cell, and leads to dimerization. In this case, HER2 dimerizes with HER3, a heterodimer, and that activates the downstream signaling and leads to altering profile of the cell, programming to program it to divide and grow and spread. And again, downstream, decreasing that cell division and DNA synthesis pathways where chemotherapy was working. But we have a number of different known ways to inhibit the HER2 pathway, the first of which was trastuzumab antibody binding to the outside of HER2 and inhibiting its signaling at that level. We have antibodies that bind to HER3 and inhibit the dimerization of HER2 with HER3, so different mechanisms of stopping the same phenomena. We have intracellular tyrosine kinase inhibitors, those small molecules that get into the cell and then bind in the pocket of the tyrosine kinase and block its signaling at that level. We have antibodies that are bivalent, that have an area on the antibody that will bind to HER2 and an area that will bind to HER3 in one molecule. We have the antibody drug conjugates that will bind to, like trastuzumab, to HER2, but deliver a chemotherapy drug. The one that we use now in gastroesophageal cancer is deruxtecan, the drug that's bound to trastuzumab, gets into the cell and then acts like a chemotherapy. Deruxtecan is a topoisomerase 1 inhibitor in the same class as irinotecan. This is not unique to HER2 pathway. This can be done and shown for different receptor tyrosine kinases, including EGFR, MET, and FGFR2. When they're amplified, there are antibodies and small molecules, some examples listed here, that work exactly the same way, but specific to each of those proteins. So how is an antibody working? An antibody works in a number of different ways. First, it can bind to and block the receptor dimerization, as I mentioned, and block the ligand binding to it to prevent the dimerization. They can also cause internalization of that receptor and degradation of that receptor, so it no longer will signal. In addition to that, they can elicit an immune response. They are antibodies, and the way antibodies work is that they can recruit immune cells like natural killer cells and others like macrophages through the back end of their antibody called the Fc domain. And these natural killer cells receptors will bind to the Fc domain and then be delivered directly to wherever this antibody has its lock and key. And if it's HER2, it would bind to gastroesophageal cancer cells that are overexpressing HER2. So antibodies work in a number of different ways, each of which is important. So to summarize the antibodies and how they work, we can have conventional antibodies that have the same epitope. We can engineer antibodies so there's more than one epitope, etc. And as an example then, a conventional antibody, again, is like trastuzumab. We can have engineered antibodies that enhance that ADCC component to elicit the immune response and recruit immune cells like NK cells, natural killer cells. Examples of that are margetuximab, which is built on trastuzumab. The top part is the same as trastuzumab, but the back end is optimized to enhance that ADCC, antibody-dependent cell-mediated cytotoxicity. We can have bispecific antibodies that bind to more than one epitope. An example of that in development in gastric and other HER2-positive cancers is zanidatamab, where it's binding to HER2 and HER3. We have antibody-drug conjugates that bind to, like trastuzumab, but are delivering a conjugated drug, in this case, deruxtecan. We can have bispecific ADCs, although there are not examples of that in gastroesophageal cancer. And we can have bispecific T-cell engagers, or BiTEs, that have epitopes on the cancer cell and on immune cells like T cells to bring them in the vicinity of the cancer cell to then hope that this T cell will kill it, essentially. So this is a complicated field and ever evolving, and so many possibilities, but you can see that these targeted therapies are leveraging the understanding that, one, we have abnormal expression of a protein and that we're going to target it with a unique and specific antibody and through various mechanisms, including immune component of these antibodies. So even though we differentiate between targeted therapies and immunotherapies and chemotherapy, you're getting the sense that chemotherapies are targeted therapies. These targeted therapies have immune properties. And so there is some blurring of those distinctions. We've talked about now how antibodies have multiple functions, inhibiting the signaling of an oncogene that's overactive and that the cell is addicted to. But we can also target any protein on the cell. And this is an example now of a relevant drug called zolbetuximab, which binds to claudin. We talked about the tight junction when we talked about diffuse type gastric cancer, when we were talking about CDH1 mutation and loss of E-cadherin and how the cells become dispersed because they're not being held together in that setting. But there are many proteins that hold together a tight junction, and another one is called claudin. And we now know that in the setting of where gastric cancer overexpresses claudin at high levels, that with the treatment of zolbetuximab, we can see benefit. And the way that we think that this is working, because claudin is not over functioning or doing anything abnormal that we are aware of, but rather this antibody is coming in and binding to it because there's a lot of it on the cell. And then through that FC domain, through ADCC, is recruiting immune cells to the vicinity to help with cell kill. So this is an example of an antibody that is really working as a targeted therapy, but by eliciting an immune response. There is, I should say, an abnormality in this gene in a rarely, mostly in diffuse type subgroup, that there's a fusion of this gene. And I mentioned it earlier in this presentation in one of the slides, that you can have a claudin fusion. And so whether there's a difference in efficacy, how well this drug works in those patients that have an actual genetic abnormality of claudin versus the majority who just have overexpression of a normal protein, there's no genetic abnormality, is yet to be seen. Another class of targeted therapies are called anti-angiogenesis drugs. And angiogenesis is the generation of blood vessels, or the formation of blood vessels, which cancers need as they grow. They need to have more blood vessels grow towards them to provide nutrients. And also this is how a major mechanism cancer cells start to disseminate throughout the body. And so drugs have been developed to inhibit angiogenesis, the first of which was an antibody to the ligand, the ligand called VEGF-A, or vascular endothelial growth factor. And so these growth factors, indicated as circles, bind to VEGF receptors on the cell membranes of endothelial cells, cells that line blood vessels. And this pathway, when activated, leads the blood vessels to start to grow and proliferate. And so by inhibiting the ligand, and then later drugs that inhibited directly the receptor, like ramucirumab, which is relevant in gastroesophageal cancer, we can see benefit by essentially starving the cancer cells from blood vessel formation. There are other inhibitors of VEGF receptors tyrosine kinases, namely, that get into the cell and bind on the inside, very akin to all of the other pathways we've already been talking about with HER2 and the other receptor tyrosine kinases, except these are on endothelial cells, not on the actual cancer cells. An important one that now will become relevant for gastric cancer, antiangiogenesis drug called regorafenib, which is a tyrosine kinase inhibitor in a recent study, showed benefit, and so will be on the radar now. How do these tyrosine kinase inhibitors work? We've talked about how the antibodies work, but how about these small molecules? Well, remember, when the receptors are inactive, these tyrosine kinase domains are not in the vicinity of each other and they are not signaling. However, when they are activated, there's a conformational change that activates the kinase domain and sends a cellular response. And there is an equilibrium between an inactive state of the kinase domain and an active state. And you can see the conformational change. This pocket opens when it's active, but it's going back and forth appropriately when it should. In the setting of a genetic event that overactivates this particular receptor tyrosine kinase, whether it's a mutation or a gene amplification that leads to too much activation, this is always on and it's always signaled. How it signals is it takes this molecule called ATP and gets inside of there that then leads to the signaling transduction. What a drug does is a biochemist designs a chemical that can fit into this pocket and displace ATP and essentially turn it off. And so there are a number of examples of kinases that are overactive and matching drugs that are designed to get into that pocket. This is showing an example of how the drug would fit right inside of that pocket lined by specific amino acids that are listed here as an example. G255 is an amino acid at that position that is making this three-dimensional whole. Something to be aware of, though, is that all of these kinases are in the same class of enzymes, and this pocket is very, very similar from one enzyme to the next. Not identical, but very similar. And so it's sometimes challenging to design a drug that is specific to one kinase. Any drug that fits into this pocket will fit into the pocket of other kinases as well. And so it becomes a little bit less specific compared to a monoclonal antibody, which really is specific to just one component of the extracellular component of the cell. So these drugs, again, get in there and they block that signal transduction by a decoy and displacing the ATP that should be going in there. So this is an example now of what I'm going to show you on this kinome map is how selective a drug can be. And we'll use the example first with HER2. And so these are the maps underlying here of the kinases. You can see that here, tyrosine kinase kinome. And lapatinib and neratinib, both of which are HER2 therapies. But you can see here what we're showing is the selectivity of the drug. And so if you see a big circle, it's highly potent, even at low concentrations. And little circles are less potent. It needs more drug to inhibit that specific area. And so this drug, lapatinib, is really specific to HER2, has some off-target effects in other enzymes, but it's relatively more specific than, say, another HER2 therapy, neratinib, which has targets all across the kinome. Yes, it inhibits HER2, more specifically bigger circles, but it has a lot of off-target effects. And so the concept then here is that these drugs can be promiscuous, or what we say is dirty, and they're not as specific as we would like them to be to say HER2 in this specific example. That's not unique to HER2 tyrosine kinase inhibitors. That can be the same for any tyrosine kinase inhibitor, including regorafenib, which is thought to be an anti-angiogenic tyrosine kinase inhibitor, as I mentioned in the previous slide, but it inhibits all kinds of other gene products in the kinome, as you can see its map here. This is in stark contrast to another anti-angiogenesis inhibitor relevant to gastroesophageal, ramucirumab, which is an antibody to just VEGF receptor 2. And so even though these drugs are both considered antiangiogenic agents, you can see that they have completely different inhibitory profiles. And this has relevance in terms of their tolerability in addition to their efficacy. So as an example, ramucirumab, as relatively well tolerated, it does have a known side effect on target is high blood pressure because it's inhibiting VEGF receptor 2. Regorafenib, which also inhibits VEGF receptor 2, does have that same on-target side effect like hypertension, but it has a number of off-target effects as well and is usually less well tolerated because of this. And one of the common side effects of these multi-kinase inhibitors is anorexia, fatigue, and a number of other things. Other drugs in the class of multi-kinase inhibitors that are anti-angiogenic, you can see their inhibitor MAPlase and inhibits quite a few different kinases in the kinome. And again, each of these is in similar class, but has slightly different profiles in terms of what they are inhibiting. This is yet another example of multiple different small molecules. Remember, they end in IB, so they're all small molecules, and they have different profiles across the different drugs here. So in summary, as an introduction to targeted therapies, we talked about antibodies, that we know that we have a number of antibodies in our immune system, but that we can make them specific to a given target. And we have various ways to do that. They're usually administered IV, and that they are usually very specific for a specific epitope. We have shown a number of examples of that: trastuzumab for 2, ramucirumab for VEGFR2. We have zolbetuximab in a positive phase 3 study for Claudin 18.2 that is expected to be approved. We have antibodies that are for other genetic events that aren't necessarily approved in this disease type, but are available in other cancer types. We have some antibodies that are only in development at the moment. There's a first-line study evaluating bemarituzumab for FGFR2 overexpressing tumors. We have different ways to engineer the antibodies with bispecifics so they can inhibit more than one epitope. We have antibody drug conjugates where we use them as a smart bomb to deliver drugs. In contrast, we have small molecules, which are usually oral. And although we try to be specific, I explained how it's difficult because the pocket that they're binding is very similar across the different receptors. And so you get often off-target effects, and therefore you can get more side effects from these particular drugs. And we named and showed a number of different drugs for a number of different pathways and receptors that are often overactivated in gastroesophageal cancer. So in this presentation on the principles of targeted therapies, I took you through the molecular background of understanding the genetics of the oncogenes that are overactivated and how they get overactivated, and then the two main classes of targeted therapies, antibodies and small molecules, and how they inhibit these particular proteins within the cell. In future video, we'll be talking about immunotherapies as an extension to this presentation and show that these are essentially targeted therapies, but that are targeting aspects of the immune response to help the immune system to recognize the cancer as abnormal and to eradicate it. So stay tuned for that. Thank you.